All right. Welcome back to the second day of the Productivity and Innovation meeting. So we'll be kind of our group in the morning and then a joint session with a lot more people coming in the afternoon. And so the first paper of the day is Songyuan Tang from Yale University presenting a paper on innovation and recombination. And we're very pleased to have Rebecca Dix from Stanford discussing.
Okay, so my name is Songyuan. I just graduated from Yale. I'm going to join Aalto University in Finland. And so I'm very happy to tell you about my paper, Innovation Through Recombination. Okay, so with this audience, I don't need to emphasize the role played by innovation to economic and productivity growth. So the key question that motivates this paper is what drives innovation? So around this question, there's actually a big debate nowadays. On the one hand, we frequently observe that new ideas often arise by recombining existing ones. For example, new software combine existing software, new existing libraries, new drugs combine existing drugs and chemical structures, and this observation of recombination dates back at least to Schumpeter, to his 1934 book. And modern growth theories also emphasize that the expanding combinatorial space may be the key to sustained economic growth. But on the other hand, other people argue that novelty is important because it can expand the stock of ideas and sustain recombination. Intuitively, with a fixed ingredient stock, then the scope of recombination can eventually decline. Especially in this era of AI, which is itself a powerful combinatorial technology, people have the concern that we might shift away from from developing novel principles, novel ingredients, towards the perhaps cheaper and easier combinations. So the debate and the development of this literature has been largely theoretical and qualitative, treating this distinction between novelty and recombination mainly as a metaphor. Whereas empirically, we don't really have a great measure to distinguish between these two, and we know little about how much each margin contributes to growth and aggregate innovation. And going one step further, taking all these into consideration, what is implied for innovation policies? Should we capitalize on the existing principles, ideas, ingredients, and subsidize the recombination of them? Or should we subsidize the discovery of new principles? In this paper, I approach these questions by making three contributions. Firstly, I propose a direct measure of novelty versus recombination based in the pharmaceutical industry. I'm going to show you that by standard pharmaceutical chemical terms, drugs are by their nature combinations of existing and new ingredients. And this can naturally divide innovation into two separate margins, the discovery or invention of new ingredients and the recombination of them into new products. With this measurement, I will be able to assess the importance of combinatorial growth. I'm going to document three empirical results. Firstly, recombination plays a substantial and rising role. Second, the force of recombination is also salient on the firm level. In particular, firms with a larger ingredient stocks, with a bigger combinatorial space, they actually specialize in recombining what they already know. Lastly, I document a value premium of novelty, which is consistent with the view that these novel innovations containing some new ingredients can open up future combinatorial space. Building on these facts, I develop a theory that formalizes combinatorial growth. The new element in my theory is that firms are not only a collection of products, but also every firm holds a set of ingredients that represent their knowledge. And firms are going to do two different types of innovations to obtain new knowledge versus combining them to new products. The model indicates declining novelty and rising recombination that we're going to see in the data, and it highlights the intertemporal trade-off between subsidizing novelty versus recombination. I'm going to skip the literature for the interest of time. And for the rest of the talk, I'm going to introduce into more detail about how combinatorial innovation works in the pharma industry. And I'm going to document three empirical results, which motivates my theory. Okay. So as we know, the pharmaceutical industry is highly innovation intensive. In the US, it accounts for more than 30% of total manufacturing sector R&D. A typical pharma innovation is a drug to prevent, alleviate, or cure a disease. In this paper, I'm going to narrow down my focus on a particular group of drugs called small molecule drugs. These drugs account for more than 70% of FDA approval and more than half of the global pharma sector revenue. So I'm also going to focus on these drugs because they give us a natural way to define ingredients and combinations. For example, this is a molecule of acetaminophen, a typical small molecule drug. It's the active ingredient in fever relief like Tylenol. For any small molecule drug, they can be decomposed into two parts, functional groups, circled in red, and carbon backbone. The functional groups are the key players. They are the part of the drug that combine with the enzyme, protein in human body. So this paper is written by one of the leading scientists of Novartis. Functional groups determine the intrinsic reactivity of the molecule and form foundation within modern medicinal chemistry. This motivates me to define or view a drug as a combination of its functional groups. And the medicinal chemistry literature gave us this readily available algorithm to identify functional groups from any small molecule drug. Moreover, this combinatorial perspective, it also aligns with how medicinal chemists conceptualize the drug discovery process. So let's take this idea into action and see how it helps us to measure novelty versus recombination. So I'm going to show you 5 real-world drugs. Let's start with 2, and I assume they are the entire prior art. Diphenhydramine and fexofenadine, these are the typical allergy reliefs that you can find in CVS. I identify their functional groups using the algorithm. The first drug here on the bottom, it contains one functional group, and it never appeared in any previous drug. So I'm going to label this as a novel innovation. Second one here, containing 3 functional groups, each of which can be traced back to previous drugs. So I'm going to label this as we can call it a new combination or a recombination. I labeled it as combination for short. The last one here, containing 2 functional groups, each traced back to diphenhydramine. So I view this as a refinement of diphenhydramine. This group is not super innovative, and it's a very small group. To fix ideas for a drug to finally reach the market, it needs to go through 3 steps. Drug discovery, to find the molecule, then clinical trials, and FDA approval. Functional groups, they are the fundamental chemical units. They're not patentable. But the drugs are patentable, and most drugs are patented at the discovery stage. Therefore, the universe of patented drugs can reflect the subspace of explored and deemed useful ideas. So I'm going to focus on the patented drugs, and I'm going to further narrow down the analysis to anti-allergic drugs. These drugs account for around 5% of FDA approval. During my sample period, they are characterized by their simple drug structure. They experience stable development effort and significant quality upgrade. My key data set is the Cortellis Drug Discovery Intelligence Database. It's widely used in pharma industry. It's also applied in economics and finance research. This database tells me all the basic information about these drugs, what they look like, whether they enter clinical trial, their development phase, which firm originates it, and what are the associated patents. I'm going to use this algorithm to identify functional groups from any of these drugs, and I obtain a dictionary. So during my sample period, I extract around 1,000 unique functional groups from 14,000 anti-allergic drugs, and these drugs are patented by around 300 firms. Every drug, on average, has 5 functional groups. Okay, so now I'm going to apply this measurement to document the empirical facts. Firstly, let me go back to my original motivation. How much innovation originates from each margin, novelty versus recombination? Panel A tells us that the number of newly patented drugs is roughly constant over the year, around 650. Panel B, I plots the number of new functional groups embedded in these newly patented drugs, and we see a sharply declining pattern. Panel C breaks down all these new drugs into different groups. Novel, combination, refinement is a small group, I omitted. Consistent with panel B, fraction of novelty sharply declined to around 6% in 2010. On the other hand, most drugs are combinatorial, and this group accounts for an increasingly big proportion over time. Overall, it shows a pattern of steady aggregate innovation and is mainly sustained by the rising recombinations. Next, I explore whether this combinatorial force is also salient on the firm level. If it is, we're perhaps going to observe that the more knowledgeable firms, because they have a bigger combinatorial space, they're going to be more innovative, and they're perhaps going to specialize in recombining what they already know. I'm going to measure firm knowledge using the number of distinct functional groups or ingredients in their past patented drugs. And I have two innovation outcome variables. One is the number of new drugs they patent in the period, and one is the number of newly used functional groups in their patented drugs. I regress these innovation outcomes under knowledge, controlling for firm size, and period fixed effects. So the result is very intuitive. It says that the more knowledgeable firms, they patent more new drugs and also learn more new functional groups. Next, I explore whether these firms also are more combinatorial. So I run a fractional multinomial logic specification where I divide all the newly patented drugs into 4 groups. Firstly, novel innovation in column 1. And then I further decompose the combinatorial drugs into 2 further groups, column 2 and 3. Combination within firm, meaning that firm recombining the functional group that they themselves used before. Combination across firm, meaning firm using the functional group that they used before, and also those that they did not use before, but were used by other firms. So these drugs perhaps contain some more knowledge, new knowledge to the firm. The last group is refinement, and this group is small, and I use it as the benchmark so you don't see it. So column 2 and 3 are the significant columns showing us that the more knowledgeable firms tend to shift away from combination across firm towards combination within. So the interpretation is that moving from a median knowledge firm to 1 standard deviation higher, the firm is going to patent 19% fewer combination across firm and 15% more combination within, which points to a firm life cycle of early stage knowledge accumulation, learning new stuff, to later stage recombining what they already know. Lastly, if the combinatorial force, again, is salient, then we might observe that the novel innovations may be on average more valuable because the new function group inside them can open up future combinatorial space for the firm. So I measure drug value using the associated patent value, which have several dimensions: their market value, scientific value from citation, their breakthrough index. Let me use panel A as an example. So I focus on those drugs that enter into clinical trials. So again, drugs are divided into 4 groups. Using the refinement drugs as the benchmark, on average, novel drugs are 100 log points more valuable than the refinement drugs in terms of market value. Then comes combination across firm, finally combination within firm. Panel B and C using scientific value and breakthrough index as the alternative value measurement, and shows us a roughly consistent pattern. Overall, there seems to be a market and a scientific value premium of novelty, which, if we think about it, it's sort of in conflict with the previous 2 facts, which shows us that on the firm and on the aggregate level, innovation is actually becoming increasingly combinatorial rather than increasingly novel. So next, I'm going to introduce to you my theory, which makes sense of all these facts and help us with the policy evaluations. I'm going to develop a firm-level tributary model, which builds on the classic Clade-Cartan framework. The new element in my model is that beyond being a product portfolio, every firm also holds a set of ingredients representing their knowledge. So this simple modification enables the model to capture salient firm-level empirical patterns. And then I'm going to, based on this baseline model, I'm going to introduce one more assumption, which is that new ideas can coincide with existing ones. And this extended model implies transitional dynamics that we see in the data, and I'm going to use it to evaluate the policy trade-offs. Let me start with the Clade-Cartan framework. There's a continuum of firm, continuum of products. Every product is produced by a particular firm, and every firm is characterized by the number of products that they produce. The key new element in my model is that every firm also holds a set of ingredients. For example, firm A have 3 ingredients. And my firms are going to do 2 types of innovations. Firstly, combinatorial innovation, where the firm is going to combine the existing ingredients into a new product. Particularly, they're going to choose this arrival rate of combinatorial innovation, and the cost would be increasing in the arrival rate. And the key assumption is that the cost also decrease in firm knowledge, capturing the idea that for a more knowledgeable firm, it is relatively easier to find a new useful idea, new useful product. For example, firm A succeeds in combinatorial innovation and obtain a product that was, and creatively destruct a product that was originally produced by firm B, and then NA would increase by 1. The second type of innovation they can do is novel innovation, in which the firm is going to come up with a new ingredient and combine this new ingredient with its existing ingredients into a new product as well. So it has an exactly symmetric structure where the firm is going to choose arrival rate. B stands for basic research, and the cost increase in that and also decreasing firm knowledge. For example, firm A succeeds in novel innovation and obtain another product, and NA and IA both increase by 1. These ingredients can depreciate exogenously at rate Kasai, and because of this creative destruction structure, the aggregate creative destruction rate equals to the aggregate product innovation rate. I'm going to show you the value function to fix ideas. Firm value, denoted by V, depends on firm state variable, knowledge, and the product. Firm is going to choose the arrival rate of the innovation, and firm earns flow profits minus innovation cost. When a novel innovation comes, firm is going to earn one new ingredient, one new product. Whereas successful combinatorial innovation only warrants a new product. Ingredients can depreciate, and every product can get creatively destructed. This simple specification actually allows a very tractable form of the firm value, which can be decomposed into two different components: their knowledge value plus the product profit value. The knowledge value term, AI, basically captures, given firm have I ingredients right now, how many future ingredients firm can discover, how many future products firm can generate, and what's the associated profits with all of these future processes. This directly leads to three implications. Firstly, firms with no products, n equal to 0, can have positive market value due to domain knowledge, which aligns with the fact that in the pharma industry, there's a lot of startups that have no profit-generating products, but their domain knowledge is expected to generate something profitable in the future. Secondly, conditional on success, a novel innovation will bring more marginal value than a combinatorial one, precisely because they bring this additional knowledge value, opening up future combinatorial space. Lastly, if a knowledgeable firm is sufficiently good at combinatorial innovation, we're going to have this property that V is convex in I, meaning that if you have 5 pieces of knowledge and I have 2, combining these 7 pieces of knowledge generates more value than keeping them separate. So this leads to an incentive for the firm to merge or co-develop drugs. So in this model, currently, I'm not further pursuing this, but this aligns with the pharmaceutical regularity that there's a lot of mergers, acquisitions, co-development of drugs. So this baseline model, it has some appealing properties, but it's not without its limitations. Firstly, it has this stationary nature, meaning that it cannot really capture the declining novelty and rising combination in the data. Secondly, importantly here, when a firm succeeds in a novel innovation, the model does not really specify whether this new ingredient to the firm, whether it's new to the whole market, or it's merely new to this developing firm, but was actually originated by some other firm in the economy. This distinction matters because in the former case, the accompanying product from novel innovation is going to be new to the whole market, it's going to be generally novel. Whereas in the latter case, this new product is merely a recombination of ingredients that already appear in the economy. It turns out that one modification can solve both of the two problems. I'm going to further assume that new ingredient coincide with any existing ingredient with the same probability. If it turns out to not coincide, then this new ingredient is new to the whole market, and the accompanying product is going to correspond to precisely novel innovation in the data that we define in the data. If this new ingredient does coincide with something existing, then this ingredient is merely new to the firm, and the accompanying product is merely an across-firm combination that we see in the data. So the difference is that originally in the baseline model, I only need to keep track of how many ingredients each firm has. But now I need to keep track of the aggregate pool of unique ingredients of the whole economy. And over time, this pool is going to expand. And then naturally, a new ingredient is going to be more likely to be a rediscovery rather than be something that's genuinely novel to the whole market. This naturally introduced the force of transitional dynamics into the model, where the measure of new ingredients is going to decline. And precisely because of this, the return to doing novel innovation is going to decline. That's why the aggregate product innovation rate is also going to decline. But it's going to be bounded away from 0 because there's abundant combinatorial opportunities that firm does. And panel C breaks down all these product innovation into 3 groups, just as we did in the data, where novelty is going to decline and cross-firm combination increases, and within firm combination also increases a little bit due to general equilibrium effects. So the model suggests that it's recombination that can generate sustained economic growth, but it does not really downplay the role of novelty, because through novel innovation, firms generate these new ingredients that sustain their ingredient stock, which sustains recombinations. Lastly, I will use the model to evaluate some policies. In real world, policies often differentially subsidize novelty versus recombination. And here, I'm going to use the model to evaluate policies that subsidize each type of innovation separately. The way it works is that whenever the firm achieves an innovation, it's going to receive a cash reward from the government. And to keep them comparable, I keep government budgets fixed across policies and compare their outcomes. So notably, from baseline to novel innovation, we can see that the novelty subsidy is going to generate the biggest boost to the growth rate, whereas it declines fast in the long run because of this phasing out and novelty getting harder to find force. Whereas the subsidy to combination is going to generate a more persistent boost to growth because of the abundant combinatorial opportunities. To conclude, this paper is motivated by the question about what drives innovation, whether it's recombination or it's novelty. And I approach this question by proposing a new direct measure of novelty versus recombination based in the pharmaceutical industry. I document that recombination plays a substantial and rising role on the firm and aggregate level. On the other hand, novelty is declining, but it earns a value premium, potentially because it opens up combinatorial space. I build a theory that formalizes combinatorial growth in which firms are characterized both as their knowledge and also as the products they're producing. It indicates that novelty is going to decline in the long run, but it is precisely novel innovation that helps firms to learn new stuff, that sustains firms' ingredient stock, and thus recombination and growth. And the model highlights the intertemporal trade-off between subsidizing novelty versus recombination. Lastly, I would like to thank the program committee for including this paper, and thank you for attending. Thank you very much.
Our discussant will be Rebecca Dix.
Okay, I'm very happy to be here to discuss this paper. So to dive in, I think this paper is super easy to motivate, as recombination is basically everywhere. Automobiles themselves are combining thousands of sort of individual components that might have gone through extensive R&D processes. Cancer treatment might be combining multiple sort of individual therapies together to provide effective treatments. And even software would be using a wide variety of sort of imports or helper packages to the point where maybe we find ourselves in a position like this, where all modern digital infrastructure might rely on some particular project that's, quote, being maintained by a random person in Nebraska, thanklessly, since 2003. Okay. So once you start thinking about innovations, sort of unpacking their underlying components through this lens of recombination, this immediately raises a number of really important questions about private and social returns to R&D that motivate the analysis in this paper. So first, what underpins incentives to introduce new components? Who bears the cost of creating reusable components? And importantly, can the innovator capture value from this sort of later use by other firms? And then second, what underpins incentives to recombine existing components? So then comparing these two, importantly, how do sort of private and social returns compare for this sort of initial component innovation versus this recombination? And I think it's pretty clear that the answers to these questions could be really important for informing certain innovation policies. But we might expect, the answers to these questions to vary across different institutional settings and may vary with sort of IP protections, how easy it is to protect components versus combinations, access to components, capabilities, and just more generally sort of underlying market structure. Okay, so then transitioning to this paper makes the very clever realization that we can think of small molecule drugs, individual small molecule drugs, as recombinant innovation themselves. So he went through this very clearly, but small molecule drugs you can decompose into sort of distinct, different chemical functional groups. This is one notion of an ingredient within the drugs. And we can immediately use this measure to show a couple of interesting facts in the pharmaceutical industry. So for anti-allergy drugs, about 79% of new patented anti-allergy drugs are just sort of recombinations of known groups, and about 13% introduce an actually new one. And we can start to try to understand, differential value of these different innovations by bringing in these sort of patent value measures, citation breakthrough scores across different types. Okay, I'll take a step back just for a minute. This is obviously a highly specific measure. In fact, it's so specific you can't even use it for biologic drugs, for example. But I don't mean that as a criticism in any sense. By being so specific, by being this very specific chemical measure, I think this is actually a really clever way in this setting to start to think about a cool way to think about the difference between basic and applied science. We can spend more time learning about sort of the introduction of particular functional groups and tracing them, their diffusion and use across different research. So in this discussion, I'm going to talk a little bit more about how we can maybe take more advantage of some additional data to trace out this sort of basic versus applied distinction. Okay, so I wanted to go through sort of a few questions that were popping up into my head when I was reading the paper. So we saw this very striking fact that the number of new functional groups being introduced for anti-allergy medications is falling over time. I wanted to think through a few explanations of why. So first thing I had to convince myself, maybe you guys were thinking this as well, but just first, is there a literal finite opportunity set here? Maybe there are only just 100 functional groups to be had. I think I convinced myself the answer is no. I won't go into the details, but if you disagree with me, let me know. Okay, a second thing is, well, are there sort of changing incentives over time that's making just using this accumulated knowledge more attractive? Or are we seeing that innovators just over time are less likely to capture sort of all of the value created by introducing a functional group? I think the answer is maybe. And then finally, is there something else going on? Are there just certain indications for which we've sort of reached therapeutic area maturity, there are shifting opportunities, or is there something measurement going on? I think this also could be part of the explanation. But of course, whichever of these is dominating would help inform a potential policy response here. Okay, so as is now, I think, classic in a lot of these innovation discussions, I want to talk through an example of how one of these functional groups was actually discovered and sort of made its way into medicinal chemistry. Okay, so let's zoom back to 1949. The Research Association of British Flour Millers was having trouble with some toxic flour that was causing seizures in dogs, and they needed to figure out why. And they ultimately discovered a new chemical functional group. Sulfoxamine was the root cause of this problem, and they published a paper in Nature. This came out of their cereals research station in St. Albans, identifying this functional group basically for the first time in the scientific literature. Okay. Then, not much happened. This functional group was largely neglected for decades. So much so that sort of a disgruntled scientist at NBER wrote a review article explicitly calling sulfoxamines and another functional group neglected opportunities, and that maybe medicinal chemists are just too focused on using groups that they've used before, and maybe we need to sort of look more broadly. This is a very, well-worded piece. He talks about two functional groups here. Boronic acid, apparently chemists were saying only a moron would put boron in a drug, and that the thought of using sulfoxamine in drugs would lead to sneers and raised eyebrows. Okay. Zoom forward, it seems like people took this to heart, and actually today there's experimental therapy for lung cancer, ceralasertib, that is actually using sulfoxamine. And then we got another review article saying that sulfoxamine is now a fashionable functional group. Okay, so this example is actually not hard to find at all. So it seems like there's a lot of interesting sort of diffusion going on. And I think we could use a lot more data to try to understand how a number of different margins might be leading to declining novelty. So the first stage here is sort of discovery. Here, some of the research that was going into discovering this functional group was not really happening in pharma. It was happening in the cereals research station in St. Albans. And so it would be interesting to sort of trace through how these new groups are entering scientific knowledge, and maybe we see underinvestment here because of this classic issue of sort of basic versus applied research. A second margin here is like the first step of diffusion I'm thinking of, which is how did we actually make the jump from cereal to medicinal chemistry? And here we need to think about how sort of access to the functional groups, how do we pay attention to them, and how do particular capabilities lead to diffusion. And then a second sort of step of diffusion here would be, well, how do we actually decide these, how do we decide to use these in particular therapies? Okay, and I do not expect you to include these margins in your model at all, but I think this could be sort of a very cool thing to think more about in the future. Okay. So what's going on in the model? Why is novelty falling? So this is sort of the extended version of the model that he showed you. Well, as the knowledge stock grows, we have this function here that's essentially saying it's going to be harder to actually find an idea that's new to market because it's going to be coincidental with another firm. Or when we go across firms, there's going to be some probability P that lowers the probability that you can actually form an across-firm recombination. And this is sort of a stand-in for maybe sort of some information blocking. You can't really patent these things directly, but you can form claims over them that might prevent other firms from adopting a particular functional group. Okay. Slight step back. If any of you want to use functional groups for your own research, they're actually, this ERDL algorithm is like super easy to do. And within like an hour, I could replicate sort of the key results of the paper. So on the left here, I'm just normalizing the number of new functional groups over time for anti-allergy medications per 100 candidates, and then I'm showing you the raw counts on the right. I did this because I wanted to understand if there was sort of heterogeneity across different indications, importantly as a function of how much we've learned about particular indications. So I extended this to a bunch of other indications as well, and we see somewhat similar patterns. For the normalized results, this is showing you results across cancer, antibacterial, diabetes, depression, hypertension, and we see that the number of new functional groups basically per drug introduced is falling. If we look at levels, we see a somewhat similar result, but there's a notable exception for cancer. And maybe this is the sense in which therapeutic maturity actually plays a really important role here, where we've just seen a huge amount of investment in cancer research in recent years, and maybe this is actually leading to an increase in novelty. Okay. The second thing I was thinking about was whether, moving a little past the model, do we think recombination alone can sustain growth in this industry, or is it maybe a way to mainly produce products? So importantly, this sort of mapping between these recombinations into actual, value. For simplicity in the model, you have a constant flow profit of different value. So it's sort of the same increment to firm profits for different types of innovations. And this is sort of motivated by the results seen using patent data. If you wanted, you could consider just bringing in additional data that's pretty easy to bring in for pharma. We could think about showing how, for example, progression across different clinical trial stages changes, or even entry into clinical trial changes. You could think about approval rates. You could think about bringing in sales data, or even if you wanted a more granular measure, think about health effects of some of these drugs, and try to understand whether in this limit, whether we might actually be approaching sort of a Me Too limit as we only go to recombination, or whether sort of the additional value of novelty is necessary. Okay, and then a last comment here. I wanted to think about sort of who benefits when we resubsidize recombination in the policy counterfactuals. So the policy counterfactuals here are these fixed budget counterfactuals where we're either trying to subsidize novel research across firm combinations or within firm combinations. And to get at this question, I think it's important to understand, once we start in particular subsidizing across firm combinations, which firms do we think would actually adopt certain functional groups? So importantly, getting back to this diffusion question of how these different functional groups might diffuse across different firms. Right now, the model is very homogeneous, very fairly so for tractability. There's essentially this common probability about whether you would actually form a particular recombination with a functional group. But I think we could all think of a lot of potential margins of heterogeneity here, where diffusion might vary sort of with firm group, fit, access, capabilities, etc. Okay, so again, I have this data on my Dropbox, so this was super easy to compute. We could try to think about using sort of other similarity metrics that have been used in this innovation literature, namely this Kriegerly measurement of the Tanimoto similarity of different molecules. You can actually apply that to just functional groups rather than entire drugs. And so what I'm showing you here is essentially the effect of a 1 standard deviation increase in chemical similarity on adoption rates of outside firms' drugs. And so what do we see is that if there's a functional group that's essentially more similar to functional groups that I've used in the past, I'm much more likely to actually adopt them. So this suggests, a little bit of homophily in the diffusion of these functional groups across different firms that might be important to sort of take into account when we're thinking about potential policy responses to, in particular, subsidizing across-firm recombination. Okay, so those are my comments. Just to sum up, I think this is a very interesting paper that introduces a really clever measurement that we can importantly use to think about this distinction between basic and applied science. But I think we can do a lot more here to push on, to try to understand further incentives to introduce novel functional groups themselves, and again, understand this distinction between basic and applied science. Thank you.
Okay, please raise your hand for questions. Pierre.
Can you turn on the mic? You should also repeat.
I think I'm set.
Please repeat your compliment.
It's a great paper, but the standard for discussion has been raised, I think. And I don't know, I'm ambivalent about that fact, maybe. I'm just, in terms of the facts that opened the paper that were kind of motivating it in a really interesting ways, it was alluded to, but I wasn't sure to what extent the firm level evidence kind of reflected what we know about kind of the division of innovative labor in this industry. And in particular, in some sense, if we think about the market for ideas between kind of big pharma and, smaller biotech firms, in some sense, do we have a market for ideas or a market for functional groups, right? Are those innovations kind of heavily relying on completely new components, right? Are they in fact brought forth by new firms in the economy or by traditional firms? And I think that distinction as empirical purchase in this industry specifically.
Thank you. Aubrey?
Interesting paper. And I have just a very quick question. So the market value that's calculated here seems to be at the patent point, but I wonder if you don't think you'll get further information if you could do sort of an event study, finance-style event study at the introduction or approval of these drugs and learn more from that, then run it, the type of drug as the independent variable and other controls on the abnormal returns.
Caroline?
This was a really interesting paper, and it was really cool to see the innovation and measurement. I had a question, I think, related to some of the results that Rebecca was showing in her discussion. So I think she actually showed that this result that you find sort of within class novelty is declining over time. That seems like actually sort of a result that replicates. But I was curious if there's also the emergence over time of new classes. And so by choosing to focus sort of within class, you're possibly missing that as sort of new classes emerge, maybe that's where the novelty is coming from, and that could sort of change the slope of the time series potentially over time.
Stanley?
Fascinating papers and discussions. My question is actually related. I think timing is quite important. So 2 observations. One is not all discovery, new functional groups are equal. Some perhaps are breakthroughs. And second, so maybe first incorporating some of that could be relevant, particularly when new classes are introduced. And second, I keep thinking perhaps timing in terms of policy response could be relevant too. Whether you should subsidize more when a new functional group is coming, when a new breakthrough comes, or maybe you want to, when the class is materialized, you want to perhaps boost new frontier, and maybe some investigation on that would be useful.
Cheryl?
Great paper, as everyone has already said. So a couple of thoughts. One is thinking about the structure of knowledge generation in the pharmaceutical industry and the distinction between big pharma and biotech companies, which are often developing these novel drugs, not to ever bring them to market, but with the hope of an acquisition. And so kind of thinking about where these innovations are coming from, so sort of similar to what Pierre was saying in terms of the market for ideas, because I think that has implications in your findings. And then also just building on what Rebecca was showing in terms of the sulfoxamines and thinking about novel implications that show up in very disparate places. And this is something we see again and again and again in pharmaceuticals. So if you were able to see, for example, with some of your new functional groups, do they show up just in allergies? And, you know, in your sample is allergies, but you could easily look at, or somewhat easily maybe, look at, do they show up in other therapeutic classes? Do they show up in FDA trials in other classes? And that's very common in pharma. So just kind of pushing a little bit further, but great paper.
Great. Scott, and then Adam.
Great. So great paper and wonderful discussion, wonderful presentation and discussion. So I had three really brief comments. So the first is, I think it's useful. I mean, I know it's implicit, everyone gets it, but sort of right. Of course, by construction, all small molecule innovation is combinatorial because there's this thing we learned about called the periodic table, and it all kind of is downstream from there. You know, and Petra Moser and Fiona Murray did some early work on that, and then Petra has a great paper. You know, before the periodic table, everything you produced, it was just like a list. You know, like, you know, what we would now view as extremely similar things, where in human knowledge, had, you know, one turned out to be red and one turned out to be blue. You had no idea coming from. So I think it's useful to sort of think about just everything must be recombination down to the bottom if you have the right physical principles. But a different way of saying that is you make the very nice point that there are layers of recombination, and I think that'd be very useful. The second is I think it's so smart to use this kind of combination of functional groups. I just think that's a really, really nice approach to it. I think you can even get further, and your footnote 7 is the key. Is Lipinski's rule of 5, which I had heard about many years ago. I don't know how. I mean, I do know how, but that's longer than I have, right? But that's sort of like that. There's these like 5 things. I can't remember. It's like 4 things, each of which is a multiple of 5 that you need to have in order to be a viable small molecule drug. That's as I understand it. It's like there's like these factors, and the function and all functional groups contribute a certain amount to those weights. Like, for example, if you can't be above a certain molecular weight and one functional group is very big, it doesn't matter how great the drug would be in principle, it's not going to be, it's not going to be usable by the human body. And so I would think that you could sort of exploit kind of a higher level of like the architecture of drug design to actually almost, and particularly with machine learning, it's a lot of combinations, but it's not an infinite number of combinations, it just seems infinite. And the last part of that is, in particular, I don't think you really, one reason that combinatorics works so well, as I understand it, is once a base, like functional groups are very contributive to safety. Like if it turns out that you figure out a way to get boron into the body, you go for it. Do you see what I'm saying? And that means you can then use that functional group again and again and again and again. Do you see what I'm saying? And then you sort of try to combine it with a lot of different things. I know it's much more complicated than that, but my like potted understanding of drug design sort of I think that has what I mean by that is if you can show a functional group is safe in one thing, it dramatically improves the probability it's going to be reasonably safe and not kill people on the way to a cure and other things. Great.
Great. Adam?
So I love this paper. I was actually a chemistry major before I was an economist, so this is like candy to me. And a little knowledge is a dangerous thing, and this is maybe just a restatement of what Scott said. I'm not sure because I haven't had enough time to think about it. But your view of recombination is very additive. And I know enough chemistry to know that probably different functional groups sort of interact with each other in determining what the drug does. So I'm just wondering if it would be worth talking to some medicinal chemists about the extent to which there might be a way to act, there may actually be novelty in choosing which things to combine. And there may actually be a way to measure that, I don't know. And maybe you're understating novelty because you're not recognizing that aspect of novelty. It's not blind recombination. They're actually thinking about what to combine, and maybe there are novelty aspects of that.
Reinhilde, and then Martin.
Yeah, just a very quick follow-up on the previous comments here. I was also wondering to which extent these new functional groups would actually matter for eventually success of clinical trials here in terms of safety or efficacy. So to which extent could you also relate your results to mechanisms of action and in order to predict that, because that's where the commercial value would actually be coming from.
Very nice paper. I was wondering whether your interpretation, at least with the model, but I think it's what you have in mind too, that the declines that you see in the data are kind of driven by the supply side of the economy, and the model is only supply side. And I was wondering, during this time period, we've also seen an increase in the demand for drugs coming from, typically becoming richer and demanding this type of goods more. And I was wondering whether that could be driving the facts and not the fact that we're running out of ways that we can recombine or novelty. And I don't know what the model would say if you just expand the size of the market, whether firms would choose to do more recombination versus novelty.
Okay, thanks very much, everyone. Maybe just take a couple minutes. I wanted to reply.
Okay. Well, so thank you very much for Rebecca for the great discussion, and thank you very much for your question and for the very detailed read of the paper, of the footnote. So I'm gonna choose. I'm just gonna have the time to answer some questions that have like a similar, like in a big general theme. So first, I find the question about it baffles me, why is novelty declining? So I have some thoughts about this. Firstly, so do we really have limited number of ideas, or here in this context we have limited number of function groups? Perhaps not, because there are so many elements, and we can combine these atoms in whatever way in whatever way that is physically possible. So there's perhaps not limited number of ideas, but whether there's a limited number of useful ideas, I think that's a question to think about. We can potentially create so many combinations, but how many of them are really useful? I don't know the answer to that question, but I think it's so secondly, an alternative explanation for the declining novelty is that there's something else that guarantees higher return. For example, there's, as mentioned, there's other fields, or there's say automation, AI, that helps more with recombination rather than developing or finding new function groups in the nature. So I would like to just put one small qualification on that idea, which is that the fact that there's other higher return fields on its own does not really mean there's going to be less and less function group discovered. It has to be that either the return to finding new function group is going to decline, or the firm is under some constraint. So I think to document what kind of constraint that the firm have, and also to document the return from other fields and the emerging new therapeutic fields is going to be something interesting to look at, and it's actually part of my future research agenda to broaden the scope of this analysis to more therapeutic areas, and to study the return of different research fields. Second, there's a discussion about basic versus applied research. So I think one of the things that makes this model different from the existing standard basic applied research framework, different from them, is that in this model, so in the standard framework, firm can do basic research, and this basic research can directly boost the productivity of many other firms. But in this model, what's different is that when one firm discovers a new function group, typically we would think of this as a basic research. Other firms cannot directly apply this function group in their own drug development. They would have to pay the novel innovation cost to learn this new thing. And this assumption is actually motivated by several facts that I see in the pharma industry. One is that we see that the knowledgeable firms, they specialize in recombining what they already know, rather than like bringing new function group in their development. And secondly, is actually what Rebecca documents, that firm tends to use the structurally similar function groups as they expand their knowledge space. And lastly, what I find is that, so I locate all those function groups that were applied again and again in future drugs. So these are the star function groups. After the first firm used this in a drug, it actually takes other firms years to really adopt this function group and patent their first new drug. So it seems that this diffusion across firms is not really straightforward. So I think this is also something that to think about for a better, perhaps better model for basic versus applied research. And lastly, I think one thing that was very interesting that was mentioned is that these function groups, they are very largely heterogeneous. Where in my model, I keep the tractability and I keep them homogeneous. So I have a future project where I study, okay, what happens when these ingredients, they have different qualities and their interactions are also pair-specific. What's going to happen? How would firm explore which combination to try first and which comes later? So I hope to be able to present the result of that project later. Okay, so and yeah, thank you very much for all the questions, and I'm happy to discuss more here.
Heidi.
Should I just go ahead? So the structure of the discussion for our last two papers needs a little bit of explanation. So every few years, we'll get submissions that are on a similar topic that we feel like would benefit from being discussed as a pairing. And so what we've done, which I think has worked well in the past, which is why we're going to try it again today, is we're going to have the two papers presented first. And then we have two discussants. The discussants are going to be discussing both papers. And so we got two people to have different perspectives, but it's not that each discussant was assigned one of the papers. So I mentioned that to say we're particularly grateful to our two discussants for agreeing to read two papers. So thank you for doing that. So the first paper is going to be Ryan Hill is presenting some work with Carolyn Stein. The second paper is going to be Mira Monin presenting some work with Josh Gans. And then our two discussants are Juan Mateos Garcia from Google DeepMind and Tim Bresnahan from Stanford.
I was going to present in the style of slam poetry, but then they turned the lights on, so I'll just do it as normal. I'm very excited to present this new project with my longtime collaborator, Carolyn Stein, about the impact of AlphaFold. So we start by thinking about science as a production process. And so we combine inputs, human labor, capital equipment, existing ideas, and recombine them to produce some outputs, which is, like new ideas. And of course, as in the broader economy, AI is now participating in this process, and there's opportunities for AI to do some of those tasks in the scientific process. And the productivity gains that we might expect from AI involvement depend on a few things. First of all, what's its performance at any particular task? Is it incredibly capable? Is it cheap? And then it also depends on the complementarity between those tasks and the other constraints in the research pipeline. And so even if a tool is extremely capable at one thing, the rate of progress might be governed in some way by these other bottlenecks that we have to tackle. And so that's kind of how we're going to think about this and study AI as task automation and how it impacts the pace and direction of science, and then also think about how researchers are adapting to the arrival of these highly capable AI tools. And we're going to do that in a very sort of notable setting in science, which is structural biology, one that's near and dear to our hearts already. Structural biology is the study of 3D shapes of biological molecules, in particular proteins. So previously, researchers had to do these very expensive and challenging lab experiments to determine the structure. They're too small to see in a microscope. And so this was something that was a challenge, and we had specialists trained to do this. Why do we care about these structures? First of all, understanding the structure is relevant to understanding the function of the protein or how it operates in a cell. And then it also can be relevant for applied research. If you're designing a drug to target a particular protein, it's useful to understand its shape. You can see that here as an example at the HIV protease. By understanding how the protein folds, you can target it with a particular compound that will bind to it. So recently, there's been a new tool called AlphaFold that was introduced to do automated structure prediction. So AlphaFold is an AI system developed by Google DeepMind, and it can predict the structure, how a protein folds, without doing any of those experiments. It's all computational. It was released in 2021. It was sort of like this very impressive and exciting announcement. It's been widely adopted by scientists, and it was even awarded a Nobel Prize in chemistry in 2024, suggesting its impact. You can kind of see on the right here two protein structures that have in green the experimentally determined structure that was done in the lab, and then in blue the predicted structure laid on top of it, and you can see basically they just line up very closely. And so in that sense, AlphaFold is very good at its job. So the insight we're going to exploit is that AlphaFold is this concrete example of AI automation for a known task in science, and we can sort of track very easily what the impact has been and how people are using it. Okay, so I'm going to focus on 3 research questions today. First, how is AlphaFold impacting the experimental structural biologists? Those whose efforts were to create these structures in a sort of manual way now have this new AI tool that really does a lot of what they did before, and so are they continuing to do it? Are they changing course? Are they adopting the tool? And so we'll focus on that. Second, we're going to think about how AlphaFold is impacting the broader areas of protein research. So for scientists who are not solving structures themselves, but might be interested in protein function or disease relevance, has AlphaFold sort of impacted them and accelerated their research? And we're going to find evidence of that and show that there's new attention towards newly illuminated proteins for which we previously didn't have any structure at all. And then we're going to look similarly downstream at drug design and medical research and see if AlphaFold is further impacting applied therapies and other patents. Okay, so there's a lot of literature, of course, about the automation in the labor market, and in particular empirical and theoretical studies of AI. I wanted to point out that there's actually a number of other papers out there about the impacts of AlphaFold, including by those in this room. Carolyn and I know a lot about simultaneous discovery in scientists, so we're very comfortable with the fact that others are working on this. Okay, so I'll start with some background, describe the data, but then I'll get right into our results. So let's go in the days before AlphaFold. How did we solve structures? Well, it was very annoying and very expensive. And there were specialists that had a lot of training to do this with methods like X-ray crystallography, cryo-electron microscopy, where you're actually manipulating the biological samples in the lab and creating data, interpreting that data, and producing a structure. The methods were expensive, very specialized training, took a long time, a lot of luck. And so unlike other tasks like gene sequencing, we never sort of made it cheap. There was no real returns to scale in this process. And so there was sort of, it ended up being a constraint for a lot of progress because of the challenge of the methods. It would cost over $100,000 to just solve even a single structure. But for a long time, it was thought that this could be done computationally. Why is that? Well, every protein is just a string of amino acids. And that sort of two-dimensional sequence folds up into a structure. And so if you have a whole bunch of input sequences, and we have some structures that have been determined experimentally, it felt like some algorithm could sort of interpret how the folding happens in a way that we could predict. But it proved to be a very complex and intractable problem for many years. Actually, all the way back in the 1990s, we had a contest every 2 years called the Critical Assessment of Structure Prediction, where they would hand computer scientists structures that had been solved experimentally but not yet published, and ask them to predict the folding structure. And you can see on the right that we weren't very good at this. If you sort of score these models by their goodness of fit, we were hovering around 30 and 40, and it sort of plateaued in our progress. Then in 2018, Google DeepMind entered this contest for the first time, adapting some of their neural net algorithms from other settings like playing board games. Turns out that's also, they were very capable at producing protein structure predictions. And you can see that the sort of quality of the structures that they were predicting went up very noticeably by 2020, reaching the point of accuracy that was sort of similar to experimental accuracy. And this was sort of shocking. It was surprising to everyone that it was this capable. Okay, so in terms of timeline for our sort of treatment for our paper, we're going to treat July 2021 as sort of the arrival or the availability of AlphaFold, both because they released the model to the public and they also created a database of predicted structures. Over the course of a calendar year, Google basically, DeepMind took all of the known proteins and predicted them, their structures, and then posted them on the internet. And so, you know, we also think of this, though, as an ongoing treatment in that there have been further algorithms that they've been introducing, AlphaFold 3 and other organizations as well. And so we see this as a growing participation of AI in this field, and that's good to have in mind as we go forward. Okay. On data sources, this is all public data. So we get protein structures that were predicted or that were solved experimentally from the Protein Data Bank. The structures that were predicted by AlphaFold are in the AlphaFold database. And then we use two sources for outcomes on follow-on research from Uniprot, which is all of the publications about a protein, including things about function and disease, have been curated there, and a similar thing for patents for applied innovation related to medical fields from a source called Schur-Kemble. Lastly, Carolyn and I recently did a survey of 427 structural biologists, and so we have some new results on that, hot off the press, that I'll include in the talk. Okay, so let's get into kind of the results of the paper. First, we're going to focus narrowly on the impact of structural biologists. Again, there's many people that were very specialized and trained to do experimental structure prediction or determination, and now we have this substitute that enters from AI that could potentially change how they work. One question you might ask is, like, did we just stop spending our time and money doing the experiments because the AI predictions are good enough? We find that that hasn't happened. So if you just plot descriptively the number of new experimentally determined structures in the PDB before and after the introduction of AlphaFold, which is the dotted vertical line there, we see really no decline. If anything, an increase in attention. So there's continuing effort to solve experimental structures. And then you could ask, well, do we have demand for those in the press? Are we publishing papers about these structures? We are. They're still being published in journal articles. And in fact, they're still being published in very good journals. So if you take just Science, Nature, and Cell and count how many papers are reporting on new experimentally determined structures, we're still doing that. And of course, those editors are very motivated to put the most important and relevant research, and so it seems like the experimentalists still have a lot to do, which is interesting. So we want to think about why that is. Like, why didn't AI just displace this field of research? In the paper, we talk about three potential explanations, which I'll touch on briefly, and you can read more in the paper. And there's sort of these three things. First is about reliability. One is that scientists might still value experimental structures to confirm or correct the AI predictions. Second is adaptation. So the entrance of a capable tool might cause scientists to sort of shift their direction and maybe work on some proteins that are more challenging for AI. We see some evidence of that, although I wouldn't say it's overwhelming. And then the third is complementarity, which is that AlphaFold facilitates more efficient experimentation, helps us push the frontier in a way that I'll show. So just a couple of results on each of these. First, on reliability, this is from our survey. We asked these structural biologists basically whether they trust the AlphaFold predictions, or do they trust it enough to skip experimental validation. Most of them say they don't trust them, all right, and they want to in some ways validate what the AI is finding. Second is on adaptation. We ask whether they're shifting their direction. It turns out that AlphaFold, especially the earlier generation, was good at a particular, sort of type of protein and conformation of proteins. But there are other ones that we might care about. So there's some proteins that have low prediction confidence due to sparse data. They also have complex multi-entity structures, proteins bound to other molecules, proteins with mutations. There are many things that might cause us to try things that AlphaFold is not as good at. And so in our project, we defined two types of proteins that are basically easy or hard for AlphaFold. The confidence scores are below above 80, whether they're multiple entities or whether they have keywords that suggest that it's a complex structure. We find descriptively not much evidence that there's a shift in attention. So if you see in red, these are the new experimental structures that would have been hard for AlphaFold. We see a slight uptick after 2021, but it doesn't really jump off the page. And in terms of the ones that are easy for AlphaFold, in fact, there's a bit of a secular decline in attention from the experimentalists. So that may be going on. We did ask the scientists about this. Do you agree or disagree? AlphaFold 2 and related advances in protein structure prediction have caused me to shift my research focus toward experimentally solving structures that are predicted poorly by AI methods. There's a lot of disagreement here. Many of the scientists say basically, I ignore it. It doesn't affect my project choice at all. I'm just doing what I'm doing. But there are a number of them that say we are sort of adapting, looking at what the AlphaFold is pretty good at and trying to do other things. Okay. And lastly, on complementarity. So it turns out that even before AlphaFold, the experimentalists were using a tool called molecular replacement that helped them to interpret their experimental data. And basically, they would take a similar protein structure and use that as a template. It's a starting point that allows them to more easily interpret their experimental data. And so in the past, you'd have to just rely on other structures that you hope were similar enough to be a good template. Now, after AlphaFold, you can use predicted structures as a template. So you can go to the AlphaFold database, pull off that data, and use it to help you interpret experimental data. And it turns out they're definitely doing that a lot. So if you look here, these are the structures in red that didn't have any other homolog. There was no other previously solved structure to start from. And after AlphaFold, we see a large increase in the use of this templates tool, molecular replacement. We can also actually see in the PDB that they report using AlphaFold as the starting point. And so this is clearly complementary to the work that they're already doing. So put all together, we think that one reason we're still doing experimental structural biology is because AI is helping us do it very well and efficiently. In fact, we asked finally the scientists, do you expect to continue doing this? We asked them, how many structures are you solving per year in the past, now with AlphaFold, and then projected into the future? And you can see that they expect to solve more structures in 5 years than they are now with crystallography and cryo-EM, suggesting that complementarity. Although if you ask them about what % of your lab's time is going to be spent on these tasks, if anything, they expect it to go down a little bit, maybe also to the story of complementarity and rising efficiency with the help of AI. Okay, now in the last part of the talk, I'm going to focus on the impacts of AlphaFold more broadly. So let's again think about scientists who are not doing structure determination themselves, but might find the structures to be useful. They're consumers of these models. So I'll start with an example here. This was highlighted in Nature magazine. This is a biochemist named Andrea Pauly, who was working on a project to understand how fertilization works in zebrafish. And there were some proteins involved. One of them is called bouncer. And she didn't have a structure of bouncer. No one had solved this before. And she wasn't trained in experimental methods herself in crystallography to solve it. So she was sort of stuck on this project and was kind of waiting around hoping someone else would solve the structure. And it turns out that AlphaFold then facilitated a breakthrough. She can go look up that protein with the computational prediction and see how it fits in with the other proteins in the process. And that unlocked some new knowledge. It allowed her to publish this Cell article about fertilization in zebrafish. And again, this is something that sort of is opening up new opportunities potentially for the broader field. So we want to look at that more systematically. And we're going to do it with a very simple difference in difference that I can describe here. So first on the left, think about before AlphaFold. More than half of the sort of proteins that are relevant for humans and disease had not been solved experimentally. So there was a whole bunch of proteins that were just sort of empty knowledge. We didn't know what they looked like for various reasons. Either someone hadn't taken the time to do it or there were real constraints to solving it experimentally. Now, after AlphaFold, we have this new set of structures that have been predicted. And of course, every protein has been predicted, but the ones that already had a structure, this represents a very small change in our knowledge stock. We already had a good model. But for those that were previously unsolved, now we have these new insights that we can take advantage of. And there might be a lot of low-hanging fruit now for people to go and make progress with. And so we're just in Difference in Difference going to compare research attention and patenting for proteins that were previously solved before AlphaFold and those that hadn't been solved, with the new shock of knowledge coming from the unsolved ones. Okay, so just a simple, difference in difference plot here, we see that after AlphaFold, there's this impressive shift in attention towards proteins that we didn't have a structure for. And they're studying things like function, they're studying disease relevance, they're studying expression of proteins. So there's clearly like a lot of opportunities that people are taking advantage of. And we can see that even these new papers tend to cite AlphaFold, suggesting that that's in many ways facilitating this shift. And so we think of this sort of as like this floodlight of knowledge that has been turned on, and so especially in those darker areas, a lot of new insights to take advantage of. We can do the same thing with medical and drug innovation. Here again we have the HIV protease, and we can look at the patents that cite that protein or mention that protein in the patent, and then we can link that in the Schur-Kemble data to each protein and run sort of the same regression with patents as the outcome. Here we don't see a massive shift in attention. So in contrast to basic research publications, patenting has at least been lagged or has not had the same shift. It appears that we're not sort of running out to make new patents about previously unsolved proteins. If anything, there's a little bit of a tick up in the most recent year, but the magnitude is about an order, quite smaller compared to the publications. Okay, so how do we think about the comparison between this massive shift in basic science, but not in applied R&D? There's sort of two potential explanations we've thought of. One is that AlphaFold is illuminating many unknown structures, but it's possible that those are not very interesting proteins. I mean, in practice, like for 6 decades, we spent a lot of time trying to solve the structures of the most disease-relevant proteins. So it's possible that the ones that are not solved are just not that useful. There are a lot of people that would push back on that view, that there are opportunities that we should be going out and seeking for more, sort of novel targets. On the other hand, it could be that new medical research is just bottlenecked by a lot of other parts of basic science. Having the structure of the protein is really not enough to sort of commit billions of dollars to pursuing a new target. There's lots of other things you need to understand about the process in the cells and how it relates to disease and how you might target it, toxicity, side effects, clinical trials. All these other things in the pipeline are still binding in many ways, even though we've automated one particular step. And so in that sense, maybe it will just take time for us to seek new therapies on these other drugs. So just to conclude, in the narrow domain where AI most plausibly is substituting for humans in experimental structural determination, we haven't seen a lot of evidence for displacement. Maybe this rings true of other parts of the economy where AI is extremely capable, but it's actually facilitating a lot of new work as well. Second, beyond structural determination, we do think it's having an impact. A lot of people are taking that new knowledge off the shelf and putting it to good use. Although we haven't seen that to the same degree in more applied areas, at least yet, and that maybe we're slowed down by other particular bottlenecks. So thank you very much. It was very exciting to present to you, and look forward to the other talk as well.
Can I use this? Okay. Hi. Well, let me start off by thanking the organizers. It's great to be here. The broad question of this project that Joshua and I are interested in is to understand, how do researchers respond to AI? Do they push the frontier and ask new questions, or do they in fact deepen our understanding of previously studied questions? Okay, so a priori, both directions are plausible. On the one hand, AI can generate novel predictions by recombining fragmented pools of knowledge and thereby allowing researchers to ask new questions. Or, on the other hand, AI in supervised learning will depend on training data, and therefore its is most valuable in questions that have previously been solved. And if effort follows where the tool works best, researchers might end up revisiting old questions. And so which direction sort of dominates is an empirical question. It's important not only because it shapes the direction of science, but it also shapes the training data that the next generation of AI model will inherit. So we look at this question in the context of structural biology, where the object of study are proteins. So you've just heard already from Ryan, but so proteins are a chain of amino acids, that's called a sequence, and they fold in a distinctive 3D shape, that's called a structure. And it's the structure that dictates the kind of function or action that the protein can take. And that's why determining or solving the 3D structure is such a fundamental and core problem in biology ever since the 1960s. Now, even after decades of experimental effort, fewer than 10% of proteins have ever been experimentally solved. And against this background, AlphaFold arrives in 2021. So AlphaFold is a supervised deep learning model developed by Google DeepMind. It was considered such a breakthrough, they won the Nobel Prize in 2024. Now, what does AlphaFold gives you? It gives you a prediction of the 3D structure. And basically, you've heard now from Ryan that basically AlphaFold threw a floodlight on the unexplored protein space. And now the question is, where on this protein space does actually experimental effort really land? And one factor that tilts the answer is that AlphaFold is most confident where data is the densest. And therefore, this floodlight could have actually been deepening our understanding of certain structures rather than expanding the frontier. So first of all, what is the protein space and how do we map it out? So you shouldn't think of proteins as isolated problems. They're related to one another through evolutionary descent. So we're going to create a sequence similarity tree, which is based on the sequence alone, which is fixed by biology, and a neighboring joining algorithm. And so here I'm giving you an example of part of the tree. It's around what's going to be the running example for this presentation, which is the human thymidylate synthase, which is a heavily studied protein that is a cancer drug target. So that's the one highlighted in blue. And so you see here in the tree that each protein is going to be a leaf, and the branch length are going to be the sequence dissimilarity, so how different they are. And proteins that are going to be sitting nearby on the tree are going to be related. Now, what does AlphaFold actually do? So, and how does it enter the experimental workflow? It'll enter in two separate ways. First, it's going to be sort of an informational shock. So AlphaFold basically predicts the structure of all proteins, but not all predictions are equal. Some are more confident than others. And so here I'm giving you the entry for a running example. And for every predictions or the 3D structure that AlphaFold gives you, it also gives you a confidence score at the amino acid level. So this ranges between 0 and 100. And you should think of this as the model's estimate of its prediction error. And so in here, you see that all amino acids in blue, AlphaFold is quite confident about, and the ones in yellow and orange, it's less confident about. And so we're going to create a measure called AlphaFold coverage, which is the share of amino acid that are confidently predicted, meaning they're above 70, which is sort of the conventional cutoff for a model to be reliable enough to use. And so really you should think of this AlphaFold coverage as a measure of the usefulness of AlphaFold at the protein level. So beyond this informational shock, AlphaFold is also sort of an experimental tool. So to turn any experimental data into a 3D structure, you need initial, like a template or a good guess of how the sequence is built or the structure is built. And so prior to AlphaFold, the template or this good guess came from previously solved sort of similar proteins, so sitting nearby on the tree. And so basically the reason for that is that similar proteins tend to fold the same way. And so here I'm showing you for our example, it's the one in blue at the top. That's exactly what happened for that protein. it was solved using as template the very nearby already solved protein related to rats. Okay, so for proteins then that are kind of far from any other solved protein, like the one in red, there was kind of no usable starting point or no usable template. And that's where AlphaFold really helps. Now it provides a template for all proteins, including those that are far away from any previously solved protein. And so this now becomes a usable template for everybody. Okay, now from the protein data bank, we can tell when a protein gets its first experiment and how many follow-up experiments it gets every year. And so basically here I'm giving you the entry of a selected few entries for our running example, which has 313 amino acid in terms of length. And so every line here is an experiment that has a year, a method, a resolution, that's how sharp the structure is, position, so this is which amino acids have been resolved amongst the 313 of that protein, and information on the depositing lab. Now, the method, resolution, and position are all related to the 3D structure, static, sort of dominant 3D structure, what we're going to call scaffold. And AlphaFold only predicts the scaffold structure. It does not tell you anything about how the protein binds or functions. And a lot of experiments actually go beyond the static structure and try to understand how AlphaFold, how different states it can take, and whether or not it binds to other molecules, which is important for drug discovery. And so we're going to tag for each experiment some of the sort of objectives or aims that it reaches. So it can be scaffold, mechanistic, so that's if it goes beyond the scaffold structure. And if it doesn't add anything new, we're going to call that a corroboration. Okay, so basically we've seen that AlphaFold can enter in two ways, this informational talk and as a template. In the paper, we have a model that shows how these two forces affect or changes a lab's decision on which experiment to run. And basically, these two forces pull in opposite direction. On the one hand, substitution basically will pull towards deepening the existing stock of knowledge. So AlphaFold provides the scaffold structure, and any experiment that tries to reestablish it becomes redundant. And in the model, experiments are, are the most valuable when it's the first experiment at the frontier, so sort of in sparsely solved areas. And that's where basically the most substitution will happen, and that's why this effect, this channel, will pull towards inwards in the sequence space. Complementarity, on the other hand, will actually push towards frontier expansion. So AlphaFold provides a template, and this template now basically lowers the cost to running an experiment, and this cost saving is going to be the greatest for proteins that are in sparsely solved neighborhood where there's no previous template to be had. And so that's why kind of the complementarity will actually push outwards in the protein space. And so we bring this to the data by basically creating a protein year panel where we track experiments run on over 500,000 proteins between 2017 and 2025. We're going to be looking at, more specifically, the number of experiments and number of follow-up experiments using a Poisson, and then a hazard of a first experiment occurring using OLS. Now, like I said, our treatment variable is this AlphaFold coverage, which is the share of amino acids that are confidently predicted. This is determined at the protein level, and it is fixed over time at the AlphaFold release. We include protein year fixed effects in order to capture features that are inherent to the protein, and year fixed effects to control for absorb common aggregate shocks. Then this beta here will really tell you how the post-2021 change in the outcome differs or changes across AlphaFold coverage in a continuous difference in difference. So I'm really comparing high AlphaFold coverage to low AlphaFold coverage protein. Now, a common concern would be that high AlphaFold coverage protein are somehow special. They're more studied, more scientifically tractable in a lab, and they're more heavily, maybe clinically valuable. And so we're going to include a set of pre-AlphaFold features that are meant to capture both the cost and the value of running an experiment and interact that with post. So what do we find? We find that actually high alpha-fold coverage really attracts more experiments. And in terms of magnitude, you see that the number of experiments increases by about 41% more for fully covered protein versus minimally covered protein. This is based on the full panel of protein year. And given the protein fixed effect, Poisson will drop all proteins that have no outcome variable that doesn't change during our time period. Then we see that in column 2, that basically fully covered protein experience a 0.1 percentage point less likely to get a first experiment. And given the baseline, that's about a 44% drop. Now this here is based on the at-risk pool. So these are proteins that have never been experimented on before 2017, which is the beginning of my time period. So this is really evidence of the substitutional channel working, where basically first experiments generally deliver the scaffold structure, and now AlphaFold delivers this for free. In column 3 here, this is on the pool of proteins that had already gotten an experiment before 2017. And we see here that the number of experiments run on those increases by about 64% more for fully covered protein versus minimally covered protein. Now I'm showing here kind of the event study plots related to those. You see that prior to 2021, not much is happening, and then there's a rise in the number of experiments for highly confident protein. Most of this is driven by follow-up experiments, and on the hazard of first experiment, things look pretty flat, if not slightly decreasing. Now we're really interested in understanding what kind of experiments are being run, and so we split the follow-up experiments by the objective or aims that it reaches. So you see that the number of scaffold experiment increases more for fully covered protein than a minimally covered protein, where most of the action is on mechanistic experiments. So these are experiments that don't just try to understand what's the shape, the 3D structure, but how the protein works. And you see here that basically a fully covered protein, you see an increase about 98% versus a minimally covered protein. And this is really where complementarity dominates, because AlphaFold only gives you the scaffold structure, and so there's no substitution effect here that should work. We also see an increase in the number of corroborating experiments, so AlphaFold now gives you a target that's worth validating. Now, I told you that the initial question was to understand where in the protein space does experimental effort actually land. To do this, we're going to create a measure called distance to the frontier. So distance to the frontier is going to be measured both for solved and unsolved protein. It's going to be the tree path distance from the protein to the nearest solved other protein. And so, for instance, in our running example, the one in blue, its nearest solved protein is the one related to the mouse in purple. And so adding sort of more structure there would be filling in the map or filling in the interior. Whereas if you look at, and so for fully covered, already solved protein, really distance to the frontier captures how sparse the neighborhood is. For unsolved protein, like the one in red here, you see here that the distance to the frontier is still the mouse, but it's much further away. Have an experiment there would be pushing the frontier outwards. And so we're going to interact, basically this distance to the frontier as a triple interaction, where basically the gamma here will tell you how the response to the AlphaFold coverage switches from deepening to frontier expansion. And so these are really our main results. What we see is that AlphaFold really attracts experiments towards the interior. You see that the AlphaFold coverage gradient is steepest near already solved or next to already solved protein and fades with distance. In column 2, you see that actually entry shifts inward. So basically the AlphaFold coverage basically does not induce first experiments to occur at the frontier, at the edge, but rather in the interior. And this is really exactly what our model predicts in terms of substitution effect, where AlphaFold provides you a scaffold, and where it destroys the most value is on first experiment, far from the frontier. And then finally, we see in column 3 here that deepening sort of moved outwards, where the AlphaFold coverage gradient is positive everywhere, but if anything, steepest at the frontier. And so here you see that basically complementarity working, where AlphaFold provides you a template that is most useful when you're in a neighborhood that's sparsely solved. So since the AlphaFold-induced experiments are going to be the training data for the next generation of AI model, we wanted to understand what kind of data are we getting from these experiments. And so now we're disaggregating even more, looking at the type of data points that we're getting from these experiments in terms of, is it the first experiments run on a cluster? That's column 1. Is it the first time we see this 3D structure ever? So a novel structure, that's column 2. And column 3, 4, and 5 is more about sort of better understanding how the function moves and binds. So column 3 is a new state that it can take. 4 and 5 is being able to bind with another molecule and how strong that bind is. And so you see that the AlphaFold coverage actually is negative for column 1, 2 in these novelty measures, but positive in the other 3. So really, AlphaFold is giving us information on more how proteins move and bind and less on these sort of novelty measures. So I think I'm okay on time, but if I were to sum up the project in one line, I would say that, here you have an instance where AlphaFold really steers where science is going and where scientists search, which proteins labs try to focus on, and not just how much it's been worked on. And so we see a redirection of experiments towards high AlphaFold coverage, and a lot of this gain is on follow-up experiments on already solved protein, rather than when you see instead that the first experiments on not yet solved protein at the frontier is actually declining. AlphaFold also pushes sort of effort deeper, not wider, so towards solved protein in sparsely solved neighborhood. It shifts the aim towards mechanistic understanding how the protein moves and binds, and we see fewer instances of sort of map broadening experiments. So here, this is a sort of a setting where we can see that AI built on human curated data can really entrench already existing knowledge faster than it can expand. Thank you.
Great. Our first discussant will be Juan Mateos Garcia from Google DeepMind.
Hello. Thanks so much, Ben and Heidi, for inviting me to discuss these papers. I really enjoyed reading them. I'm very excited about the discussion. And before I start, I should flag that I'm not a professional economist. I'm just a humble data analyst at Google DeepMind who works in a team who spend a lot of time looking at the adoption and impact of our models, and hopefully that brings some insights that will be relevant for the discussion today. I just wanted to start by saying that this research is amazing. It's really the right stuff, the kind of study that we need to be able to understand the impact of AI in science. If our starting hypothesis is that AI is a general purpose technology for science, and I'd love to hear what the team thinks about that, then this means that it improves fast, that it's being adopted pervasively, and that it generates innovation complementarities. The adoption of AI looks very different in mathematics, to biology, to economics. This means that we need very timely research about adoption of AI, looking at different fields, and really paying attention to these deep dynamics of adoption and impact. And this is exactly what these papers do. So thanks so much to the authors for taking the time to do this deep and insightful work. Just wanted to say something very quickly about AlphaFold. We had excellent introductions to it, but I guess I just wanted to capture this idea that AlphaFold is many different things. AlphaFold is a data set, 200 million protein structures that any scientist can go and use, but it's also an open source tool that scientists can use to generate new predictions about structures, for example, structures in different situations, structures interacting with other molecules and with other proteins. And finally, as we have heard, AlphaFold is also a tool that helps to interpret experimental data and provides almost like a prior or a starting point for experimenting that data, making experimentation more efficient. So, what do these papers do? So Monen and Gans start by modeling the reallocation of experimental effort, very much focused on what's happening with biologists who run experiments to generate new protein structures. I'm going to be giving a bit of a caricature of the paper because I don't have a lot of time, but I guess the way I think about it is almost about whether AlphaFold is impacting on experimental exploration or exploitation. Exploration here is discovering new structures. Exploitation is deepening the understanding of existing structure, those mechanistic insights that Myra mentioned. And basically what's happening here is that AlphaFold lowers the costs of all experiments. So in principle, it's beneficial for both exploration and exploitation, but it actually competes with experiments that are more exploratory, because why bother running an experiment to create a structure when AlphaFold is already giving you one? But it actually complements this deeper exploitation work, which involves analysis or entities that AlphaFold is not able to predict. And therefore, what this means is that we should see labs shifting away from exploration and into exploitation. Their approach is basically to track the position types. So you have an experimental structure submitted to the protein data bank. So is it an exploratory one? Is it an exploitative one? And the treatment, like we heard, is the prediction confidence of AlphaFold. Like Mira said, how useful is AlphaFold going to be as a replacement for this experimental structure? Findings very aligned with the hypothesis, an increase in exploitation for proteins exposed to AlphaFold. And this is particularly the case for proteins where a lab already had previous experience. It's more like a sense of perhaps lock-in going on there, and also a decrease in the positions for novel proteins. And I guess the concern here is that this is decreasing the diversity of data for future models. My takeaway is that an event deployment landscape shaped by previous history can turn a floodlight into a street light. Looking at Hill and Stein, the focus is, they also consider AlphaFold impact on experimentation, but I would say the focus is much more on what happens downstream in biology and also translational impacts as biological insights become technologies and therapies. The approach is again like building a wonderful data pipeline that considers experimentation and publications and patents. And the treatment is whether we have a structure for a protein before the arrival of AlphaFold. And the idea here is that if we have a structure, then the arrival of AlphaFold doesn't change much. But if we didn't have one, this is new information that biologists can leverage. So what do they find? They find an increase in experimentation for the proteins that lack close templates, because AlphaFold is providing those now. And also we see this big increase in experimentation, in downstream biological research, focusing on proteins for which we didn't have experimental data before. And as Ryan said, this is almost showing us how AlphaFold is enabling experimentation and research in less well-mapped areas of the protein universe, the proteome. Looking at translational impacts, actually we have weak evidence of translational outcomes for now. Maybe we're starting to see something starting to happen in the most recent year, and this points at the existence of scientific O-rings that could block or slow down the AI impacts, and this is even before we get into clinical trials. This is just like about the research to develop an understanding of a biological process that might lead to the development of clinical trial candidates. So that's the summary of the papers. I did a bit of additional analysis inspired by this work. And basically what I did is two things. First of all, I looked at how scientists are actually using AlphaFold. It's a toy analysis. I really looked at a sample, but what I did is take papers that cite AlphaFold, work with the full text, extract summaries of how these papers used AlphaFold, and then cluster those descriptions of the use cases to build a taxonomy, which is what you can see on that side. And it's almost going from very aggregate segments of activity into more disaggregated ones. I think it's very interesting that most of the activity is about applications and it's about biology research. It's not about experimentation. It's only like the thing at the bottom, the purple category that is capturing the use of AlphaFold for experimentation. Most of the activity is in the downstream biology uses of AlphaFold. And only consistent with that, only around 10% of the papers within this data set are actually generating PDB structures like experimental data. The other thing I did was look at deposition types used in downstream, basically a citation analysis of deposition types to see who's citing different types of depositions, who's citing the exploration, who's citing the exploitation, just to get a sense of, it's almost like what is the different types of value that are being created by different types of experimental analysis. Okay, so now I'm going to get into the discussion. So the first comment is, I'm sorry, it's super boring, but it's like data and AlphaFold diffusion lags created challenges for these kinds of timely studies. I didn't want to lean too much on this comment or criticism because I really want this research to happen. I don't want us to wait. But yeah, still it's worth flagging. And we have data lags, which is you need to crystallize, collect, analyze experimental data. You need to publish. The data needs to get annotated. Patents are filed. Patents are published. So that's going to create a lag. And maybe a bit more interesting, like for this audience, is there also like lags which are related to the process of diffusion of AlphaFold as a new technology for structural biology. And basically what's happened here, and we see this with all of our models, the model gets released, and for the first few months, it's just validation, benchmarking, scientists kicking the tires of the model, figuring out what it works, what it doesn't, what it can do, what it can't do. And it's almost like the community is building trust about this model to decide whether they want to invest on this model and bring it into their workflows. Another diffusion lag comes further downstream, which is when we have looked at citations to AlphaFold citing papers, we find that actually initially, like a lot of that activity is fundamental biology research, like a more basic research, and then it's only when you start to get into those indirect impacts and indirect influences when you see like more medical research starting to appear. And I guess this might be a particular issue for unsolved proteins where there's a weaker evidence base about the biology of that protein that might create additional lags in generating understanding, building understanding of that protein and then turning that understanding into an application. So what are the implications of this? One of them is potential for right censoring, and I think this could be a particular issue for Monen and Gans, which is that perhaps at this point we are seeing the easier depositions. There may be the depositions that are closer to the frontier or more aligned to what the labs were already doing. And the question here is, have labs had the time to explore or pivot towards the frontier? For Hill and Stein, the question is more about, do you need to wait for a bit longer to start to see those translational outcomes? They mentioned this really nice study by Matteo Tranchero that shows actually like translational timelines of around 4 years for new gene and disease pairs, which would suggest that maybe we should start to see something for AlphaFold, but then gene disease pairs feel to me much more of established technology and much more applied than something like AlphaFold, so maybe its translational timelines are a bit more compressed to what we get from AlphaFold. Okay, then my second comment is about the fact that AlphaFold versatility might make it quite difficult to define a clean treatment. If we look at Monen and Gans, they talk about, I think Mira, you said, AlphaFold coverage determines how useful predicted structure is. But actually, when I look at my AlphaFold citations, I find quite a few examples of AlphaFold being used to solve protein structures where it has low confidence predictions. And the reason for this is that scientists are, for example, using AlphaFold to identify what are the weakly parts of a structure and then use that to modify the experimental data in order to run the experiment. And this suggests that AlphaFold might also be useful, not just where it has good coverage, but also where it has bad coverage. And this might lead to a change in the interpretation on the new deposition or exploration coefficient. In other words, it's not just that we are seeing a decline in depositions for new structures where AlphaFold has good coverage, but also an increase in the depositions for proteins, for new proteins where AlphaFold has bad coverage, because it's helping with that kind of research. With Hill and Stein, it's almost like the flip side, which is that we have seen from Monen and Gans that there's a lot of work that seems to be enabled by AlphaFold, which is follow-up work and it's incremental work, and it's just building mechanistic insights of previously solved proteins. And actually, I looked at the types of citations that those types of depositions get, and I find like there's, for some of these categories, for example, validation, we see like lots of citations coming from medical research, which suggests that actually this might be a pathway to translational impact being generated by AlphaFold, which is not through the treatment of this is a protein where we didn't previously have structural data. Just to conclude, my last comment is about how do we interpret these findings. And I guess one from for Hill and Stein is how do we disentangle AlphaFold impact from scientist choice. We have like DeepMind throwing 200 million protein structures at scientists. Maybe many of them were not solved before. How do scientists actually choose which ones to tackle next? What is that decision-making process? And understanding it maybe help us to disentangle what is the impact of AlphaFold from the behaviors of scientists and the decisions they make that shape that impact. And here, I mean, I just wanted to flag that the authors are in an amazing position to think about potential explanations and mechanisms through the amazing work that they have done in racing dynamics, also in protein science, but also, for example, think about pivot penalties and how they might shape how scientists respond to the big shock in the availability of protein structural data provided by AlphaFold. I feel like my comment around interpretation to Monen and Gans is that I feel like the framing of the results is very negative. Like it's almost like AlphaFold is reducing exploration, AlphaFold might be narrowing the availability of data for future models. It's almost consistent with this, to me, cliche that AI is bad for scientific creativity. And actually, the important thing to remember here, paraphrasing Halt and Kochfire, is that protein structures are not the thing. Protein structures are the thing that take us to the thing, which is biological understanding and understanding of disease and application. And actually, it might be that we are pushing that frontier of biological understanding with AlphaFold. As I said before, 90% of AlphaFold using papers don't involve PDB depositions. This is biology research that's using AlphaFold. And also we find when we look at the citations received by different types of depositions, that actually some of the more incremental ones tend to receive lots of citations from basic biology, and also just tend to receive more citations full stop, suggesting that AlphaFold is pushing frontiers in useful ways. And if we want to consider comprehensively and fairly the impact that is having on biology and exploration, we should also consider those types of impacts. So, concluding remarks, just to finish, how do we go beyond AlphaFold? This research is amazing, it's super useful, it's very insightful. But the question is, what else do we need to be looking at? This diagram here is just a list of models developed by Google DeepMind or Google that are relevant for science. And this includes models in more AlphaFold models, not just AlphaFold 2, AlphaFold 3, AlphaFold Multimer, AlphaMissense, models in genomics, models in physics, in mathematics. We have specialist models, we have LLMs, we have agents. Some of them actually complement AlphaFold. Maybe when agents start to be able to do some of that biological analysis downstream, it accelerates things. So how do we encourage more research looking at these models as well, so that we can start to build up an evidence base about their adoption and impact as robust as what we are getting for AlphaFold, thanks to the efforts of Mira and Josh and Ryan and Caroline. So yeah, I guess my conclusion is that there's a lot of work to do in this space. I'm looking forward to seeing, hopefully, research on all of these models being presented in future editions of this workshop. Thank you.
Thank you, Juan. And next we'll have Tim Bresnahan.
Well, we have two super cool papers on an extremely important problem. We have, how do you get this thing to go? Come on. Thanks. Yep, I got it upside down. Right. So when I teach the economics of innovation, I always trip over the projector cord in the second lecture. The papers are about an extremely important topic. Blessedly, they talk very little about the labor market patents. They talk about the output of the science. That's really good. They go way down into the weeds, and I want to really thank authors for teaching me about, what is a scaffold, things like that. And then they have this commonality that they have to deal with AlphaFold being both a complement to and a substitute for experiments. So that's one really interesting area. And then they go after different efforts to deal with follow-on research. And that produces a lot of knowledge. And they did a good job. Juan did a good job. So I'm going to confine my attention entirely to unfair questions about, what we might learn in the future, with maybe some grumps. So my interpretation of the substitution and complementarity is knowledge, at least in this field, is contained in literatures, not in experiments or papers. And by the way, you might say that about this literature too. We have another author. Where are you? At least one more in here. So if you want to talk to people about this stuff, you can talk to Jerry as well. What's the dominant scaffold? That's one of the several scaffolds that it might have. That's what AlphaFold's about. There's really good evidence about the complementarity part, which is maybe a surprising part because of this, the use in molecular replacement. Okay, and so my first sort of unfair question would be, these papers are about the impact of a big drop of scientific results on other scientific work. And it would be harder but useful to see whether that's different from a big drop of experimental work. So that would require, because of the enormity of the AlphaFold shock, I'm just going to shut up about econometrics, right? You need a good instrument to get to how is AI different in this impact from, or these impacts, because these guys study a lot of things, from experiments. But we don't have that. That would be an interesting thing. Okay. So that's going to give you an idea when I say it's all going to be unfair. We're going to do more of that. What about the complementary stuff? They pretty much told you, the authors and Juan did a really good job of explaining all this, all the things on this. The one thing I want to say, my one econometric remark, this variable which in the paper is called dist was F in Myra's slides. That's the distance to the frontier from the protein at hand. The frontier solved by experimentalists the year before. There's an extremely careful examination, approximately on page 70 of the appendices, of robustness to definitions of that. So this, I'm going to argue with them, as one did, about the interpretation of depth versus breadth, but it really seems like there's a fact there. Okay. Now, continuing, I sort of have two kinds of easy complaints. The first one is, and this is a thing that shows up in the paper, but it just shows up as a weakness, a worry about potential selection bias in the previously studied experimental things. But it's also an important analytical point. You should think that the linear model is false. There's multidirectional causation between knowing about, for example, functions and knowing about the structure. And we know from, probably people in this room know a lot more than I do, that coordination across those disciplines is quite hard. Downstream in the pharma companies, even within the firm, it's quite hard. Absorption is quite hard. So, and I think this is just me saying in different language, one of the things Juan said, in the short run, the ability to coordinate around such a huge drop in knowledge may be limited by how hard those coordination costs are, right? I mean, this is sort of agreeing with the authors. What we're talking about here is a complementarity and coordination problem. And so that could be, here I'm exactly following one, right? That could give you something about the slowness that we see in one paper and something about the depth over breadth we see in the other. That, and so this is sort of an optimistic complaint, I think, right? In the short run, you might see this slowness and this narrowness. And again, here I'm also closely following one, but using a very different language. The short run supply curve of science, especially when it needs to be coordinated, is probably rising pretty steeply. And we see, now here I need two different underlying truths to have complaints about the two papers. Right? You could have, if there's racing across labs, well, that could explain, the depth, because people race where they already know where they're going to get hit. And if you have the, or on the other hand, if it's difficult to pivot, I'll use unfair language again, right? You could have the slowness in the other paper. Okay, so now there's a grump that I do every summer. I know people are going to be sick about this. I have this grump about all papers of the form, does X influence Y, or how does X influence Y? AI is a huge object, collection of objects. Not all of them are like one another. The papers study a big drop of information, okay? That's what the AI did here. It might do lots of other kinds of things. And so, there's some thinking about external validity. I'm going to agree with us for a minute. And then there's some other things which aren't external validity, which I want to talk about a little bit. So there are folks out there who are trying to make something like AlphaFold 2 for much bigger molecules, like for DNA and RNA. Okay, that's sort of like this. The kid, I think of her as a kid, although she's a very distinguished senior scientist who bought my house when I retired from Stanford, is trying to make AlphaFold 2 for cells. She thinks Emma Lundberg, she thinks AlphaFold 2 is the bee's knees. She's like a macroeconomist to AlphaFold 2 is microeconomist. She really likes it, but there's a quantitative model of every protein that's in any particular cell you want to solve, and you don't have to wait for the micro guys to get off, measure the elasticities you want to know. So that's where external validity seems to be the right word. But then there's all this other stuff, there's all these other new tools. The one that came to my mind reading these papers was like information retrieval from other disciplines. Automatic library functions were right after inverting matrices. That was the second use of computers in science, way back in the early 50s. AI is dramatically better at that. So, that could help with some of these coordination problems. There's the thing that we don't see here. Any new theories? There aren't any new theories. There aren't any new conceptions. That huge drop might produce that. And then all the stuff in applied microeconomics, which is sort of radically different than when we saw, just to take the other science we all in this room know a lot about, we just learned a lot about biology. So, this is sort of an interesting case that's at one extreme, right? It's this big drop of new knowledge, right? There are lots of other cases, and so that would form my interpretation rather than worrying about external validity. Okay, so I grump that every summer. you'll fire me soon. Okay, and the last one here is the most unfair of all my unfair questions. Right now, what historical moment are we in the history of science? The external environment funding has gotten dramatically worse. Funders, both by composition effects and by changes in the behavior, have gotten dramatically bossier. So what's the interaction between AI and science interacted with that change in the environment? And my first thought was it's sort of is it a cost reducer or a opportunity expander, because we're about to come under intense cost pressure. And so you could interpret Myra and Joshua as saying it's a cost reducer, but forget for a minute whether that's an over-interpretation. So that might be a happy finding for our moment in history because the funding's going away. But then there's also this increased directiveness of the external environment, peer reviews going away, all that stuff, which might put different values on there. In other words, I'm saying a different version of exactly what Juan said, like raises more questions than it answers. Yeah, that's a really good feature in a paper, and in this case of 2 papers. Thank you.
Did you say what?
So thank you to authors and discussants for a wonderful session. So I wasn't a scientist before an economist. Okay, so my prior going into this was that AlphaFold is this incredible, miraculous thing that's going to make human life way better. And I guess taken together, the evidence is that there's huge reallocation of effort towards things that AlphaFold makes easier for scientists, and kind of no evidence of an increase in outputs. So am I wrong that my posterior now is that it's not as obvious as I thought that AlphaFold is going to be kind of good for humanity if we're just kind of reallocating effort with not producing more kind of quality outputs?
John? John, can you check that mic's on?
How's that?
Good.
Okay. So compliments to the guy for the papers. I had a comment on Ryan's paper. I was really interested in the surveys that you were doing of scientists and how they were responding or not responding to some of the things that were going on in AlphaFold. I wondered if you dug into a bit on the heterogeneity between the different answers. So, one view might be that the older incumbent scientists are not responding very much. But the real response, we see this in a number of different areas, comes from the new entrants. And so you could look at that by looking at the kind of, by age, looking at the younger scientists and seeing whether they were the ones who actually were reacting to the new opportunities. And it's really the older guys, some of us in the room might feel that way, who are not responding in the way that they should be responding.
If I were writing this paper, I would actually start to understand impact, where impact comes from. I would start from citations, citing this paper. What kind of fields? What is the context in terms of how it's cited? Maybe it's cited as it doesn't work. Or it's cited, we cracked a lot of protein folding with this. Or maybe you'll find Tanya Babina citing this paper as an example in her economics paper of AI, right? So I think it would be interesting to understand how, where impact is. And small alternative mechanism for lack of results is maybe experimental structures, no decline in experimental structures is driven by the fact that journals still required to do this experimental confirmation.
Martin and then Jerry.
These are 2 fantastic papers. I've seen Caroline present very early versions of this at Berkeley, so I'm happy to see how it evolved. I think the 2 discussants, both Juan and Tim, pointed to something that I think is important. So Juan mentioned that AlphaFold is not only one thing, it's 3 things. And then Tim mentioned this idea of knowledge versus of this being just a drop of knowledge. And I just want to give sort of like my lens on that. And I completely agree with those points. And I think what the reason for that is that you can think of these papers as saying, oh, what happens when you have just a drop, an increase in the stock of knowledge? And I think if that's the interpretation of the papers, then that tells us something about the return to scale in the innovation function. If I think about models of innovation and growth, that's what this is telling us. And it would identify that parameter in these models. But if I think about AlphaFold and what these papers are doing, and I don't think this is the right interpretation, but if I think of it as a tool, and the true notion of what an AI tool is, and not just knowledge, then I think it's identifying something different. It's identifying how efficient is the investments in innovation in creating new knowledge. And it's a completely different parameter in the models that we use. And given the timeline, my guess is that what you're identifying is the knowledge effect and not the use of AI as a tool. But I think both papers will benefit from kind of like making that distinction and telling us like what is it that we think we're identifying here.
So first of all, I'd like to thank the opportunity of the Innovation Research Bootcamp, so a PhD student like me can join the discussion with two groups of people, scholars that I admire a lot. I have 2 points for each group because I also work on this question. For Ryan and Carolyn, I think my first point is that, I also see the complementary of AI through the molecular replacement in X-ray crystallography method, but I think that's only on the intensive margin, but on the extensive margin, I also think it's also important to note that the aggregate growth of knowledge is mainly driven by the capital-intensive cryo-EM method, which are good at solving the proteins that has a poor prediction. Number two is that, yes, I was very excited to see all this publication increase, but I think it's important to understand the welfare implications of all those new entries into the field of biology. For example, like, are we actually building things on false positives because we have so much AI tools and people don't bother to check with whether AI said is actually ground truth or not? And for the other group, Mia and Joshua, I think my biggest question is, I don't know, like the restrictions on your sample to only include the Swiss prot database before 2017, which includes around 500,000 proteins, but that's like the whole protein sequence is much wider than that, and it seems that the idea search space is also wider than that. And also I think the dynamic interaction is very important. And maybe also important is to think about where the training data is happening in the private sector, like the pharmaceutical firms, or versus is still happening in the public sector, which is academia. And thank you so much for your wonderful presentations and discussion today.
I'd like to echo this. Thank you to the authors for these wonderful papers and presentations, and for the discussants, for the interesting discussions. So I have one question mostly for Ryan and Carolyn. This is about the complementarity result. So I wondered if it's particularly protein structure experimentalists who start to do more complementary work to the kind of information and new insights that AlphaFold provides. But could you see if perhaps other scientists, perhaps in the biomedical sciences, are now jumping in? So what I'm wondering mostly is, do you see that the experts start to do more complementary work, but more novices, the ones whose bread and butter is not protein structure, they start to do work that's related and that's now enabled by the information, the insights that AlphaFold provides? And then I also have a question for Myra and Josh. I wondered about your AlphaFold coverage variable. As I understand it, this is related to how much information, how much data AlphaFold has to base itself on, and that determines to some degree the reliability of their predictions. And so I wanted, this data presumably is the result of where investments, attention has been made, so presumably where there is a big payoff to understanding a protein, but also where experiments have been successful. And so I wondered if you could try and do some more work in teasing those two apart. So particularly, do we see that there's more work in the areas where AlphaFold has better predictions, simply because those were the protein structures that we were interested in the first place, and there's the biggest payoff? Or do we now also see that perhaps there's more work in areas where previously experiments were unsuccessful, and now with the help of AlphaFold, we can push further? Thanks.
Great. Again, thanks for the two great papers and two great discussants. One of the things that was thinking about is, where are these changes occurring with the use of AlphaFold? Is it happening in academic labs? And if so, can you distinguish between more basic academic labs and more applied academic labs, such as in medical schools? And then likewise, is it being implemented differentially in the corporate setting? Is it happening in incumbents? Is it happening in startups? So, and this, I think impacts both thinking about the papers and publications and patents, but also how it's being deployed in terms of this breadth versus expanding the frontier. And then, just sort of on a broader level, thinking about the sort of parallel to the introduction of polymerase chain reaction, or PCR, in roughly mid-1980s, which really revolutionized molecular biology, biotechnology, had big impact on the lab. So sort of similar to what John was saying, is that the younger researchers were all over PCR. It took much longer in some of the more established labs. So just kind of looking at who's adapting it, and who's basically arbitraging this new technology. And then again, sort of this differential impact both between academic and industrial science.
Reinhilde, and then Pierre, and then Scott.
Yep, that was a great session. My question goes to Juan. I really liked your introduction of these three different layers of publications, which you identified through text mining here, and it goes way deeper into just applications versus the rest here. It would really be nice also to be able to use this to identify, are these different layers, who are the players in these? Are they connected or not? Are they different? And would that also help us to explain, like, for instance, the time to adoption here? So would that be different for the different layers here? Also the incumbency thing here, would that be the same players that already have experience or not in the different layers, or is there more room for new entry in any of these layers here? And where the biggest improvements in terms of science eventually would be here? So it would really be nice to be able to validate that three layers here and then use it for analyzing some of the questions of the papers.
So I note with glee that on current trend, structural biology will take over the innovation meeting within 2 or 3 years, and I want to be here for it. So I'm part of the ever-growing set of applied economists who enjoy talking to structural biologists, and when kind of reporting on the effect of AlphaFold on their work, they all note that it's been a fantastic experimental companion. And one reason, I think, is what both papers talked about, which is this idea that those structures now provide a very convenient template. But there's another idea that I think is a bit more nebulous and probably harder to pin down in the data, but potentially even more important, which is that AlphaFold is a great device to generate new hypotheses. To, for example, help focus the scientists on what part of the proteins are actually worthy of kind of more attention, or deletion, because they're going to kind of interfere with kind of the vision of where the protein is going to have its impact. And I think it might be worthwhile for both papers to think about how that could leave a distinct kind of empirical signature that it could then kind of exploit, trying to think about what's going on.
Scott.
So, as somebody who is not part of the growing army of studying structural biology, so very, very interesting. I love both of these papers and this whole area, because it's clearly a very well-defined space to study this. But I want to maybe return to something that Tim said that I think is really important, which is, because so for the last 30 years, you could probably even, once you add in the PCR example, you could probably pick 6 or 7 GPTs that hit the life sciences, all of which were supposed to very quickly and rapidly dramatically expand the range of things we could do. And we did get, out of PCR, we did get CSI, and we can, do blood. And for criminology, it was very, very quick. But the rest of it was much, much slower, right? I mean, essentially, stem cells, basically, we're 25 or more years into it now, and there's therapies that build on stem cells, but definitely did not deliver the promise. CRISPR, transformative for scientists, and only recently, now we're in year 15, only recently transformative for therapy, and only in narrow cases. What I mean by that is, while I hate social media, I know that there exists this world that says like, oh, AI-enabled tools and regenerative research, regenerative research is going to dramatically transform the ideas production function so rapidly, and this resequencing. And it just seems more Bresnahan and, Trachtenberg, Bresnahan and Greenstein as it relates to adaptation, GPT. It's going to require a whole new generation of scientists to figure out how to use these tools rather than dramatic expansion of innovative productivity. And that's what I took. I think papers are great, but the idea that basically people make changes on the margin, they choose the projects that are most worthwhile to pursue, I think that's a very good canary in the coal mine of what this is telling us about these kinds of transformational tools. And I think Tim's point that we should say, what's special about AI here, and what do we more generally learn about the introduction of a massive reduction or massive improvement in what you can do scientifically through a new tool is, I think, informative.
Bill?
Very brief at a very high level. What I'm so struck by is the fact that AlphaFold exploits the existence of this reservoir of objective correlatives, of the proven X-ray and otherwise determined structures of proteins. When I think about its prediction power, that's what I begin with. When I think about the prediction power of large language models in the social sciences, what I worry about is that we tell each other again and again, the map is not the territory. But in the social sciences, it's kind of maps all the way down. Where do we find our objective correlatives against which we can gauge the prediction power of this amazing technology?
Dan.
Like those before me, I am not a structural biologist, though for a hot minute I was a structural economist in a former life. But we're not that interested in that. But picking up on something that John mentioned, Scott was getting at Tim, and all of Tim's co-authors have gotten at. I know Tim didn't want to get into the labor, any kind of labor thread of this story, but in the background, I'm kind of thinking about how our lab structure is changing. Yes, there is a recent Toronto student, Gabriella Cavalli, that I guess Myra and Josh will probably know. I'm not sure if Ryan and Carolyn do, that have put together some data to document, how the composition of labs that are using AlphaFold did change afterwards. But I think I'd be interested to see more of that woven in here, in particular in thinking about up-and-coming researchers, students, postdocs, and even young PIs. What are they learning differently, doing differently? How are those skills being brought to bear into incumbent PIs, labs, in resolving some of these bottlenecks? Is there heterogeneity within these samples, as John was getting up to younger, older PIs, or with different lab structures or skills in their labs? Part of me says this because I'm only, a dozen years out of my PhD, but I already feel like a dinosaur. With apologies to those who are older than me, how technology is changing now and how we do research. In order to get some things done, I'm really reliant on PhD students today. So to think about what that looks like in this context and how that might be part of how these bottlenecks get cleared out is maybe something one can start to look at with the data you have.
So both of these papers were just extremely thoughtful on adjustments in the scientific process, which I think is very much of interest to this group and something that's also predictive of kind of later outcomes. I did just, for Ryan and Carolyn, I think it probably won't be in this paper, but I feel like people are going to interpret a lot out of your patents graph, and I feel like if you're going to have that in the paper right now, it's very underdeveloped relative to how it's going to be interpreted by people. People are just very fundamentally interested in the question of AI and drug development. And so, just a few examples. So right now you're looking at granted patents because I assume you don't have data linked in on patent applications. You can date those back to, the application date and at least kind of think about kind of that timing. Also, were patent paper pairs kind of an important feature of this in the pre-period? And do your results basically imply that the incidence of patent paper pairs has changed? Or how do you think about that as kind of one way of interpreting the data? People will also, I think, want to interpret that as, the quality of these leads, as you discussed, might be changing, but it could also be that somehow the research that's being done is more distant from the therapeutic applications that are known. And so if there's something that you can do to kind of analyze, is the type of language suggesting that kind of there's less knowledge that's scaffolded around the potential therapeutic applications, such that you would expect kind of a longer time to diffusion for transitions to things like patents and clinical research. I think those are all things that seem knowable from the text of the data in the patent applications and kind of how those papers are kind of written. And those just seem like things that, again, I think it might be a separate project, but that might give some nuance to how the loaded interpretation that people are going to give on that one graph.
I'll also just comment, these papers are really great steps forward. I think you learn a lot from them, and I think we're all very curious about it. And but of course, we have intertemporal spillovers, and so now it opens up new avenues, and you're getting comments along those lines. And not for these papers. I think this is probably too much to take on, but I was quite taken by the kind of AI meets human behavior aspect in the discussions and in some of the other comments. And particularly, Juan said is sort of what is the choice that scientists are making. And sometimes you guys are studying that, but there's also the kind of collective choices in science, like the allocation problem. I think this is also where, Tim talked about coordination in some sense. That's the similar idea. I think there are a lot of structural biology labs, but I think there are a lot more proteins. I'm pretty sure about that. So there is an allocation problem, and it makes me think of Hugo, Oppenheim and Squintani, and herding. And you could herd because we know there's more value in certain proteins, but you could also herd because we're just engaged in sort of sociological things in the sociology of science, and everyone needs an audience, and we need reviewers and grants, and it's just hard to explore for other reasons. And so, this kind of interaction between AI and human behavior and our sort of institutional systems of science, I think is a really interesting question. And so, I think herding, for example, the kind of the collective exploration and the collective exploitation, this is really the pivots to people are, it's sort of going beyond the individual lab and thinking about that sort of broader question of where I think science is really done, not by everyone doing the same thing, but by somehow spreading out. Okay, so it sounds like we have great discussions. Okay, so I think we're coming to the end of our narrowly focused innovation program. That was not very narrow. We're going to break for lunch in a minute, and then we're going to reconvene. Oh yes, and I'm going to stop saying this in a second. Hold on. We're going to reconvene. I'll finish the thought at 12 at 1:30 after lunch. But I'm getting ahead of myself. We do have time. We should give these guys a chance to reply. Ryan and Mark. Yeah, thanks.
Ben's herding us out of the room. All right, we've had enough. Okay, I do have, these were all so thoughtful. Both the discussions, Juan and Tim brought such interesting insights from different perspectives, but all very helpful. And I really enjoyed the other talk as well. I think, like we saw, there's a lot of complementarity. Just a few things on the suggestions from the room, which were fantastic. First of all, on the sort of evidence of an increase in output versus shift in attention. That is actually something that we're trying to pin down. It's a little tricky within the data, especially because this is very recent work. We definitely see this reallocation of effort. We're trying to figure out ways to sort of document whether this is increasing total output in science. We don't see strong evidence of that yet, but it's something we're working on. a few great comments from John and Dan about the survey. We're very excited to do things like heterogeneity and get some other insights from there. I mean, one thing on Dan's point about younger generations, just to preview a little bit, we actually asked the scientists, would you recommend that your students learn how to do cryo-electromicroscopy and X-ray crystallography? And overwhelmingly, they all said, yes, we strongly think they should continue to learn experimental methods, which was pretty fascinating to us. So there's also, this came out of Jerry's comment and some others, sort of the role of experts. One interesting contrast we find in our results is that the experimentalists are very sort of cautious about taking the predictions off the shelf and running with them. They want to validate, they want to deepen, they want to sort of learn, more of the details of the structure, so they think that's important. But I will say it seems like the consumers of these, the other scientists, they're sort of just taking this stuff and running with it. And it's an interesting question about how we should feel about that. On one hand, it's great that it's opening up new avenues for research, but questions of quality and reliability do loom large if they don't have the expertise to sort of validate those predictions themselves. And then just one or two more on Heidi's point about patents and trying to understand that context more. I agree that that's part of the paper that is a little more recent and we're trying to understand better, and we'll definitely do that. The last point I'll make is related to Scott and what a few other people said about AlphaFold as a GPT and opportunities for accelerating progress. I don't actually know if we want to take a strong stand on this is what AI is doing. AI is, of course, many things, but in particular, there's this view in the world, not very popular on Twitter, but about AI as a normal technology, which is that AlphaFold has dropped a bunch of knowledge on us. It can do new things, but there have been other, moments of rapidly improving and cheaper tools like PCR and gene sequencing and so forth. In some ways, AI is doing that at quite a large scale. I liked all the different models that DeepMind is working on. We'll see if that means we are going to just get more of those tools all at once. But I will say a lot of the impacts we think likely are quite similar to other moments of tool creation and automation in science in the past, which has really facilitated the progress that we've made over time. So thank you again for all the comments. We're going to read them carefully. Thanks.
Let me chime in on some big picture things, because it's not like I've read my own paper. So the thing is, first of all, we love AlphaFold. Nobody here is anti-AlphaFold. I understand we're looking at it hard, but we're not anti-AlphaFold. Secondly, the reason why this is an exciting area is, one, AlphaFold is an unprecedented shock. Quickly won a Nobel Prize, it opened up its space, and also partly because, and what it is, it did so in a space that at last has a measure of knowledge. It has a measure of distance of knowledge. And why do we need that? Because a few years ago we got a great paper by Chris Cornell, who's over there, and Schneider, a quest for knowledge that sort of gave us a framework for thinking about science choices regarding how far and how close they move to the frontier as opposed to deepening knowledge. And that sat there in our space, especially without the focus that we had on experiments and the type of experiments going on. And at its heart, there's a theory going on here. And so what I want you to have 3 words to take away from this exercise. Le Chatelier principle, baby. The Le Chatelier principle says that long-run elasticities are going to be longer than short-run elasticities. But it says, in certain circumstances, and if you look at the model, it'll go in the same direction. So with respect to our paper, what that means is it's not a concern for is AlphaFold going to have an impact and other things going on. It is a concern if the future versions of AlphaFold that get us to 100% of all proteins in that space are relying on inputs from human experiments going on today. Because if the humans are choosing not to do the experiments where AlphaFold needs it the most, that is the issue, and it's precisely the tension that came up in the Quest for Knowledge paper. And so the canary in a coal mine, I can't remember who said it, that we are seeing is that it's not going in the right direction, and Google needs to get its act together and promote experiments in the interest of promoting that AI if it wants to see it develop. That's the bigger takeaway. And so we're not going to get a singularity tomorrow. We all knew that. No one at Silicon Valley knows it because we know that science is slow. That's all this is partly confirming. So if anyone thought differently, we get a big shock, it didn't change anything. But at the same time is to develop these AI tools, we still have work to do.
Okay, so just forecast, remember we're coming back this afternoon, and that will be joint with digitization, so it'll be even more people, for 3 more papers. Yeah, thanks for a great session so far.